The researchers analyzed biological samples from over 8,000 individuals across different age groups, tracking what are called epigenetic markers, chemical tags on your DNA that control which genes are switched on or off. They were mapping the pace and pattern of biological aging across the lifespan. What they expected to find was a gradual, linear progression, aging as a slow drift. What they found instead were two distinct acceleration points, moments where epigenetic aging doesn't walk, it runs. The first occurs around age 44. The second, more severe one, around age 60. The 44-year inflection point was the more surprising finding. In the months surrounding this age, researchers observed accelerated changes in pathways governing lipid metabolism, cardiovascular function, immune regulation, and, critically, the systems that repair DNA damage. In plain language, the machinery your body uses to fix itself begins to lose efficiency precisely at midlife. The errors accumulate faster, the repairs slow down, and the compounding begins. Your epigenetic clock is not a metaphor, it's a measurable biological reality. Scientists can take a blood sample and calculate your biological age, which can differ from your chronological age by a decade or more in either direction. Two 45-year-olds sitting in the same waiting room might have biological ages of 38 and 57. The 57-year-old doesn't look sick, doesn't feel sick, but their cellular infrastructure is running on borrowed time, and the standard annual physical will not catch it. The medical establishment isn't telling you this yet, not because the science is unclear, but because the infrastructure to act on it at scale doesn't exist in most healthcare systems. The research moved faster than the clinical pipelines. You are ahead of the curve just by knowing this exists. Here's the concept I want you to hold on to for the rest of this video. The longevity lockdown. Think of your maximum lifespan as a ceiling, not fixed from birth, but progressively determined by the decisions, exposures, and biological events of your life. And that ceiling gets locked in, set in concrete, during your 40s. Before your 40s, the ceiling is relatively malleable. Your biology has significant repair capacity. Damage is real, but largely reversible. The epigenetic markers are still fluid. After your 50s, the ceiling has largely solidified. You're managing what exists, not building new capacity. The gains from intervention, while real and important, are smaller and slower. But during the 40s, this is when the concrete is still wet. This is the last window where your interventions don't just improve your current health. They rewrite the upper limit of what's biologically possible for you. A massive shift just happened in how the longevity science community thinks about midlife. The old model said, aging is linear, intervene at any age, and gain proportionally. The new model says, aging has leverage points, and the 40s are the highest leverage decade you will ever have access to. The people who understand this are not working harder than everyone else. They're just working at the right time. And right now, the clock is ticking on whether you're in that group. Let's get specific about what's actually happening biologically during this acceleration window. Because epigenetic changes is abstract, what it looks like in the body is not. Mitochondria, your mitochondria, the energy-producing structures inside every cell, begin to show accelerated dysfunction in midlife. Their efficiency drops. They produce more reactive oxygen species, free radicals, as a byproduct. That oxidative stress damages cellular membranes, proteins, and DNA. This isn't a slow drip. The Stanford data suggests it accelerates sharply around the 44-year inflection point. The result isn't just fatigue, it's a systemic increase in cellular inflammation, the kind that doesn't show up on a standard blood panel, but that drives nearly every chronic disease of aging. cardiovascular disease, neurodegeneration, metabolic dysfunction, cancer risk. Your telomeres, the protective caps at the end of your chromosomes, shorten with every cell division. In midlife, the rate of shortening accelerates. When a telomere becomes critically short, the cell either stops dividing, entering what's called senescence, or it dies. Senescent cells don't just sit quietly. They secrete inflammatory compounds, what researchers call the senescence-associated secretory phenotype, or SASP. They poison the tissue around them. And in midlife, their numbers begin to accumulate faster than the body's clearance mechanisms can handle. You are, quite literally, accumulating zombie cells. And the 40s are when the zombie population starts to reach critical mass. Cognitive resilience. Your brain's ability to compensate for and repair neurological wear also peaks and begins its decline curve in the 40s. The Stanford study specifically flagged pathways related to synaptic plasticity and neuroinflammation as among the most affected during the midlife epigenetic acceleration. This does not mean cognitive decline is inevitable. It means the window to build neurological reserve, the buffer that separates normal aging from dementia, is closing. What you do in your 40s determines how much reserve you carry into your 60s, 70s, and beyond. Now we shift, because knowing the mechanism without knowing the intervention is just anxiety in a lab coat. Here are the 3 protocols with the strongest evidence base for intervening specifically in the midlife epigenetic window. These aren't general wellness suggestions. Each one targets a specific mechanism we just discussed. VO2 Max is the single strongest predictor of all-cause mortality in midlife adults. Stronger than blood pressure, stronger than cholesterol, stronger than blood glucose. A landmark analysis in JAMA Network Open found that moving from the lowest quintile of VO2 Max to the next quintile reduces mortality risk by more than improving from any other cardiovascular risk factor. And here's what makes this particularly relevant for the 40s. VO2 Max declines at approximately 1% per year, starting in the late 30s, unless you specifically train to counter it. By 50, if you've done nothing, you may have lost 10 to 15% of your aerobic capacity. That decline is not trivial. It maps almost directly onto your mitochondrial health. The protocol, 2 to 3 sessions per week of what exercise physiologists call Zone 2 training. Sustained aerobic effort at 60 to 70% of maximum heart rate, where you can hold a conversation but are clearly working. 30 to 45 minutes per session minimum. This targets mitochondrial biogenesis directly. It stimulates the creation of new, efficient mitochondria and slows the accumulation of dysfunctional ones. Complement this with 1 to 2 high-intensity intervals per week. True high-intensity, not just a bit harder. 4 to 8 intervals of 3 to 4 minutes at near maximum effort, with equal recovery periods. This is what actually drives VO2 Max upward. Zone 2 builds the base. HIIT raises the ceiling. The target: get your VO2 Max tested. It's available at sports medicine clinics and increasingly at consumer health labs. And aim to be in the top 2 quintiles for your age group. If you're in the 40s and your VO2 Max is above 40 min, you are in a fundamentally different risk category than if it's below 30. Muscle mass peaks in the late 30s and begins its decline in earnest in the 40s, a process called sarcopenia. What most people don't know is that the rate of decline is not fixed. It is directly modifiable by resistance training. And the muscle you build and preserve now is not just cosmetic. It is metabolic infrastructure. Skeletal muscle is the largest glucose sink in the body. More muscle means better insulin sensitivity, lower ApoB, lower inflammatory markers, and, critically, a larger reservoir of functional capacity to draw on in late life. The data on muscle mass as a longevity predictor is among the strongest in the entire aging literature. The protocol: 3 to 4 resistance training sessions per week, targeting all major muscle groups with progressive overload, meaning the load increases over time as you adapt. The mechanism of sarcopenia prevention requires that the muscle be stimulated beyond its current capacity. Maintenance-level training slows the decline. True progressive overload can reverse it. Protein intake is the other half of this equation. The research on protein requirements in midlife has shifted significantly in the past 5 years. The outdated RDA of 0.8 grams per kilogram of body weight is insufficient for muscle protein synthesis in people over 40. Current sports nutrition and longevity medicine consensus points to 1.6 to 2.2 grams per kilogram as the effective range for muscle preservation and synthesis. Distributed across meals, not concentrated in one sitting, because the muscle protein synthesis response to protein is rate-limited per meal. This is the protocol that gets the least attention, and arguably has the most direct mechanism on the epigenetic processes the Stanford study identified. Polyphenols are a class of plant compounds that do something remarkable. They act as epigenetic signaling molecules. They don't just have antioxidant effects, they interact directly with the methylation patterns on your DNA, the same patterns the epigenetic clock reads. In doing so, they can slow and in some cases partially reverse epigenetic aging markers. The most evidence-backed specific compounds for midlife epigenetic intervention: resveratrol and pterostilbene, found in grape skins and blueberries respectively, activate sirtuins, a class of proteins that regulate cellular stress response, DNA repair, and inflammation. Pterostilbene has superior bioavailability to resveratrol and is increasingly the form used in research protocols. Effective dosing in studies: 50 to 150 milliliters per day of pterostilbene. EGCG, the primary catechin in green tea, has demonstrated direct effects on DNA methylation patterns in multiple human trials, with particular relevance to the inflammatory pathways activated in midlife. 3 to 4 cups of high-quality matcha or green tea per day, or a standardized extract, achieves research-relevant serum levels. Quercetin, found in onions, capers, and apples, has an additional property particularly relevant to what we discussed earlier. It is one of the best-studied senolytics, compounds that selectively clear senescent cells, the zombie cells we talked about. Quercetin, in combination with a pharmaceutical called dasatinib in research settings, has demonstrated meaningful senescent cell clearance in human trials. The quercetin alone data is less dramatic but consistent. The practical protocol: a diet genuinely high in colorful plant foods, not as a vague directive, but as a specific daily target of 30 or more distinct plant species per week, which the research consistently associates with microbiome diversity and epigenetic health, combined with a targeted supplementation approach for the 3 compounds above. This isn't about supplements replacing a poor diet. It's about using the most direct epigenetic tools available at a time when your epigenome is at its most receptive to intervention. Let's bring this together, because the goal of this video was never to make you afraid of your 40s. It was to show you that your 40s are not a passive experience. They are an active intervention window, the most consequential one you will ever have. The longevity lockdown is not a sentence. It's a deadline. And deadlines, when you know about them, are an advantage, not a threat. The people who will live the longest, the most functional lives, are not doing extraordinary things. They are doing 3 ordinary things with extraordinary consistency during this decade: building aerobic capacity, preserving muscle, and using food and targeted compounds to directly signal their epigenome toward health. That's it. That's the entire protocol. The complexity is in the mechanisms. The practice is simple. The concrete is still wet. The question is what you're going to press into it. If you're watching this in your 40s, you now have a framework that most people won't encounter until it's too late to apply it at maximum leverage. If you're watching this in your 30s, the window hasn't opened yet at full intensity. But the habits you build now determine what your 40s look like biologically. You're not early. You're on time. If you're watching this in your 50s or beyond, the lockdown is not complete. The ceiling has solidified, but it hasn't sealed. The interventions still work. They work harder if you start in the 40s, but they work at every age. The research is unambiguous on this. The next video in this series examines something the Stanford study also flagged: the specific role of sleep architecture in midlife epigenetic aging, and why the sleep changes that most people write off as just getting older are actually one of the primary accelerants of biological age. If you think you can optimize VO2 max and ignore sleep, you're running the engine without coolant. Subscribe so you don't miss it. And if this changed how you think about the decade you're in, share it with someone who's in it with you.